Hi, this is Miss Litton, and these wonderful students are participating in the Honors Bio Unit 2 exam review. Say hi. Hi. Take two. And this entire presentation, the PDF of what I'm doing on the board right now is in your Google Drive folder for Unit 2. And I can also attach it right onto this YouTube presentation as well if you want a copy of it. There, I put a lot of information in here, so we may not have time to do all the questions, but I know a lot of you like to get the answers for the questions, so even if we don't do them, we'll get the answers. The other thing you need to know, if you are listening to this review, is this is a new and updated top 10. A little bit different from the one I presented in class, just because I needed to add some chapter 4 on there, so I combined a few, okay? And I've also put that out on Instagram and Twitter and the Remind app, so you will get that. All right, so if we take a look at each of these items on this list, you want to possibly, for yourself right now, maybe you put your name in your little group shared review notes that you've got going. Each of you sign in on your own, add your own little list to this. What are the things that you're most concerned about when you look back at these? So take just a minute to look through the list and think, what are the things I'm most concerned about? Don't just say I have to do 1 through 10. Look to see what are some of the top issues for you that you might struggle with a little bit. And tell the person next to you, I'm a little concerned about this. I'm a little concerned about this.
I just want to go. Yes.
All right, so if you picked your top maybe 4 things that you had, was anybody number 1 in your top 4? Okay, 2, 3, 4. Okay, 4 seemed like bigger. Okay, 5. Nobody. 6. A little bit. 7. A little bit, okay. 8, okay. A little bit bigger than a little bit. And 9 and 10. Okay. That way I know where to focus my time with you on what's important to you. So characteristics of life, oh, let me hit this too. Make sure you know your lab. It's only one lab. It was like a combo lab, and you just finished it. And we talked about it in class today, but I can go over that with you if you want. Do your additional objectives, so you're familiar with those. Read through your terms a couple times. Those terms should be familiar to you because they're part of what we discuss in class. Yes?
I know there was one additional objectives that didn't get on the calendar. Will we be tested on those?
If it's not on the calendar, I won't test you on it. Like when I'm writing the test, I look literally on the group portion of the test, I literally go through these and I try to pick a question for each class period, one thing from each of these, and then something about the labs. That's how I go through. So if it's not on my calendar, it's not showing up. Okay. And then your group shared notes. You definitely want to look through those. So coming back to this, we had this in our, the characteristics of life. So let's go through those together. Living things are, do it with me. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and they develop and they adapt and die. Okay? So we can nail that list, no problem. You just want to make sure you know how to kind of break each of those issues out. So for instance, living things are organized. Let's go through the organization level, and that would be atoms, molecules, organelles, cells, tissues, organs, organ systems, organisms, population, community, ecosystem, biosphere. Good. Acquiring materials and energy, I think you know you know, like we need oxygen and nutrients for us, right? We do cellular respiration. Maintain homeostasis. That's keeping a constant what? Internal environment despite fluctuations. Responding to stimuli, maybe with our nervous system or our endocrine system. Pretty sure you know about reproduce. They grow and develop. I think, you know, we have units on that. And adaptations, that was part of what we discussed, right? And we talked about the mechanism for evolution being natural selection. Good. And die. So that's all of this. Okay. We also talked about so we've done the characteristics. Oh, that's nice. Okay, so we've done let me get a different color here. We've done the characteristics, we've done the organization. Let's do classification. Okay, so if we were going to do that, we would want to talk about the 3 domains. Okay, and 2 of the domains, archaea and bacteria, have something in common. What do these 2 right here have in common? They're both built out of prokaryotic cells, right? Do you remember that? Okay, both of these are prokaryotic. Okay, and then eukarya obviously is eukaryotic. So then we want to think, okay, what's the difference between prokaryotic and eukaryotic cells, right? And we'll hit that one later when we go into cells. But as far as class, you know, one's simpler, one's more complex. Key thing on eukaryotic, membrane enclosed organelles, right? Like the nucleus. Okay. But we said we have these 3 big domains. So what makes each of those unique? Archaea and bacteria are both prokaryotic, but one is found everywhere and one is in extreme locations. What one is in extreme locations? Archaea. So they have different membranes, different cell membranes, different cell walls that probably allow them to live in those other environments. And archaea is most closely related to eukaryotes, more similar in their DNA, ribosomes, etc. Okay, domain eukarya, you want to differentiate between these 4 kingdoms. Remember, it goes domain, then what? Kingdom, phylum, class, order, family, genus, species. What's most inclusive? Domain. What's most exclusive? Species. Good. So we understand the 3 domains. So now let's just talk about the 4 kingdoms within the domain eukarya. So this is horrible to read right here, but this is protista. Okay, and then fungi. And then this is plantae or plants. And what's the last one? Animals. Okay, so I would say you want to make sure you know for animals that they are multicellular. Most of your plants are also multicellular. Most of the fungi are multicellular, but there's also some unicellular. Protista can be uni or multi. Which ones are heterotrophs?
Plants.
Heterotrophs. Animals are heterotrophs, and we're called ingestive heterotrophs, meaning we take nutrients right in. Whereas fungi are absorptive heterotrophs. And what would we call plants?
Autotrophs.
Autotrophs. And then you get both with protista. And those are like the key things you would need to know. Okay, so I think that you would be fine if you knew those. Okay, and fungus, mushroom, plants, you got that down, and we're animals. Yes?
So are those the types of eukarya?
Yes, that's underneath the domain eukarya. So if I was going to do some sort of like chart, right? So I have bacteria, archaea, and eukarya. If I was going to do a chart, okay, I would say both of these are prokaryotic, right? And these are eukaryotic. Are we good on that? We know what those mean. And these archaea live in extreme environments, and they're more closely related to eukaryotes. I'd say bacteria are everywhere. Okay, then of eukarya, they have 4 kingdoms, and those 4 kingdoms are Protista, fungi, plantae, and animalia. And within those kingdoms, there are phylums and classes and right, and on down. And we talked about man's classification too. Do you remember that? What was man's classification? What domain are we in? Eukarya. Okay, what kingdom?
Animalia.
Yeah, animalia. You're going to remember animal kingdom, right? And remember it goes domain, kingdom, phylum, class, order, family, genus, species. And how can we remember that? Did King Philip come over for good soup? Okay. So our domain that we are in is eukarya. The kingdom is animal. Phylum is chordata, PC. If you remember that, okay. Domain, kingdom, phylum. What class? What class? Look around the class. There's a bunch of mammalia, right? Look at all the mammals. Domain, kingdom, phylum, class, order, and we said what?
No monkey.
No monkey, and we remember what? Primates. Good. Domain, kingdom, phylum, class, order, family, and we said family is Hominidae. The family is Hominidae, right? And then genus, species was Homo sapien. Okay, got that? All right. So then the next part of that, we went into talking about the scientific method and how we make observations, ask our questions, do our research, and then we're ready to write our hypothesis, right? Our guess. And then we talked about doing our experiments, analyze your data, and conclusion. And remember in our hypotheses, we talked about, did we talk about research and null hypothesis? Yeah, so we talked a little bit about that. And then when we analyzed data, we looked at things like the chi-square test, right? To analyze our data to see if it was significant or not significant. Right? We talked about the different kinds of variables, independent and dependent variables. Independent variables are your experimental, and one way to remember this is they look I and E, okay, in both vowels, independent and experimental, and response and dependent variable. And the way I remember that is dependent, it's dependent on what? Data. It's your response, it's your data. Okay, then we had a bunch of questions I kind of jammed in here, and we can just kind of run through them really quickly, okay, just so we check it out here. I'll only do the ones that you can do a multiple choice to, not the, I won't take the time in the typing ones. Let's go Starfire. There's more than 13 of you in here. Clicker it. This we should get 100 on, and we did. Good job, Hulk Smash. 911 number. You know what chapter comes next, right? Atomic structure, types of bonds, water, dehydration synthesis, condensation synthesis, right? Hydrolysis, 4 important organic molecules. Shaka Khan. Okay, why don't we like D? What's wrong with D? They picked that. Molecules. Then what? Then is this okay?
Tissue, then organ.
Tissue, then organ. Good. All right. So disappointing. All right, we did that. And here, it's similarities in evolutionary history. What's the ultimate source of energy? The sun. The big yellow one's the sun. What are organs comprised or built out of? What do you build an organ out of? Tissues. Good. What is the mechanism for evolution called? Natural selection. We went over that. What's the most inclusive category? Domain. What genus is man categorized as? Homo. Good. Which kingdom contains eukaryotic, multicellular, absorptive, heterotrophs? Fungi. Absorptive fungi. Absorptive heterotrophs. Eukaryotic, uni or multicellular, they could be autotrophs, they could be heterotrophs, that is protista. Death of a species or larger classification category. Extinction. Good. Quantitative data is measurable, true or false? True. Okay, what type of variable, give both names, is the result or change that occurs due to the experimental variable? Response. What's another name for it?
Dependent variable.
What was that? Dependent. Dependent. Data. Dependent. I don't know. Have you? Have you not? Okay. All right. Now, if I go back to our master list, we did everything on 1. Okay. Now we're on 2. Okay. So, atomic structure, ions, covalent. Let's go through that. So why don't we do the atom story? Ready? The atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 1832. 8, 8, 8 is great octet rule. If we just unpack that, we kind of have a lot of the first part of the chapter, okay? So the periodic table is arranged by the number of what? Protons. Protons, okay? Now, I can figure out electrons from that because however many positive charges, I usually have the same amount of negative charges. If I don't, I am an ion, okay? Now, the atomic mass consists of what? Protons plus neutrons. So if I know the proton number, because that's the name of the element, right? So if I know the proton number and I know the atomic mass, if I know this and this, I can solve for this, right? Because this and this make this, right? So I can solve for that. When we talk about electrons and we want to put them away, we know we fill up the lower energy levels first. And I know how many electrons I have because I know how many protons I have. So the first level can hold up to how many? 2, and then 8, and then 18, 32. And we say 8 is great. That has to do with making elements happy. 2 ways to make them happy. Either what? Fill them up or get them to 8. If you don't fill them up or get them to 8, they will want to acquire some electrons either permanently or share in order to get happy. If you're acquiring them permanently, you are becoming an ion. If you are sharing them, you are entering into a covalent bond. If you share fair, it is called a nonpolar covalent. If you share unfair, it is polar covalent. Okay? And we know and why they would want to enter into that bond is because the 2 or 3 or 4 or whatever is in that particular molecule, they're trying to get stable. Okay? Now, if we talk about water, let me jump here. Let me hit some pictures because I went really fast. So we see something like helium. We know this is the mass. This is the atomic number. This is the number of protons. And we know that this mass is protons plus what? Neutrons. So 2 plus 2 would be 4. And if I was going to put them away, I would put 2 electrons in that first energy level. Yes? We could do the same thing with lithium. What could you tell me right away about lithium? 3 protons, 4 neutrons, 3 electrons, right? Because 4 plus 3 is 7. If I want to put these electrons away, it is what? 2, 1. Is lithium happy? No. Why not? Yeah. So what will lithium? He'll either share to get to 8 in that second level. You know, somebody who has 7, he can hook up with them. And then together they have 8 in a covalent bond. Or he can what? Yeah, give up 1 and become an Ion. Perfect. Okay, we looked at the periodic table. Remember we said periods are always on the what? Horizontal. Okay, and we remember that. And groups are on the vertical. And on the groups, we can see, okay, everybody in this group has 1 valence electron. Everybody in this group has 2, 3, and so on, right? And these are working out of the same level. This is all on level 1. So this one has 1 valence electron. This one has 2 valence electrons, so he's the 2. Everybody here already has the 2. This lithium is 2 plus 1, which is 3. This one is 2, 2, which is 4. This one is 2, 3, which is 5, right? You with me on that? And that's how you work your way through. So they're at the same level. And we know that you can determine how much an element will react based on the number of what? Valence electrons. Perfect. Okay. So we talked about that. We talked about that. Then to get stable, we talked about they can either form covalent bonds or ionic bonds. And then let's get into water. When oxygen binds with 2 hydrogens to form water, oxygen is really electronegative, and he will share his electrons with hydrogen, but he doesn't share fair. So what kind of bond does he form? Covalent. Yes, a polar covalent. So when we look at the bonds right here, here's oxygen, here's a hydrogen, here's a hydrogen. That bond between each of these oxygen and a hydrogen, this is the polar covalent bond. And because of that, the oxygen becomes partially negative and each hydrogen becomes partially positive. So the partially positive of one water molecule has got a weak attraction for the partially negative of another water molecule, and that is what a what? Hydrogen bond is. So because of that, we have different properties of water, like it's a liquid at room temperature, even though oxygen is a gas and hydrogen is a gas. When they hook up and they're hanging on to each other, they are liquid at room temperature. There's a good solvent. You can dissolve things in it. If it's charged or polar, who can't you dissolve? What doesn't mix well with water? Oil, fats, right? Okay. It fills up vessels. It can gain and lose heat slowly, right? So it's good. It cools our bodies. It maintains our temperatures well if we live near the ocean, because it can hold the heat during the day and then re-radiate it back out. High heat of vaporization, and it's less dense when frozen, which protects the life underneath. Do you remember those discussions? Okay. Just so much. I know I'm kind of going fast, but I want to make sure I hit everything before you leave. And for those of you who can stay, I'll make sure I go through the whole if you're here and I have somebody to talk to, I'll go through the whole presentation so you have it in case you have to leave a little early, okay? You'll have it. And then we talked about pH as well. And remember, pH is measuring how many what ions you have? Hydrogen ions. The lower the pH, the more what you are? Acidic you are. Okay? The lower the pH, the more acidic you are. But when we say low pH, it's really you have a lot of hydrogen ions. Okay? You have a lot of hydrogen ions. Okay? And then that brings us to the second part of this chapter. Okay? Do you want me to run through those questions I skipped over? Yes? Okay, here. How many neutrons are found in calcium? 20. Because you're doing 40 minus 20, right? Okay, and then this one, how many neutrons in chlorine? So you're going to round down, so you're going to take 35 minus 17, and that's what? 18. Okay, how many electrons does it have? Let's put them away. 2.
8.
Is he happy? What does he want?
One more.
Perfect. Okay. We already did all that. What's this one? Oh, we already did all that. Okay, let's go here. Ionic bonding involves loss of electrons, gain of electrons, attraction of opposite charges. All of them. Good. Atoms of an element that have the same number of protons but different numbers of neutrons. So when you vary the number of neutrons, what's that going to be? Isotope. So I know my first blank is isotope, so what can I cross out? A and B. Gone. While molecules that share electrons, so now which one am I going to pick? Yeah, I'm going to pick covalent. Okay? Good. Which of these ions is important for acid-base balance? Hydrogen. Good. Carbon can engage in how many bonds? 4. Now, we know this because it goes 2, comma what? 4. 2, comma 4, because it has 6 electrons, right? Okay. So now, with the next thing we talked about was, we talked about hydrocarbons, which are long carbon chains with hydrogens on them. But we talked about building our 4 important organic molecules and then how we go about doing that. Okay, so first of all, do you know what your 4 important organic molecules are? Yes?
You didn't say the answer to the last question.
Oh, I didn't? I'm so sorry. 4 covalent bonds, C. C. Okay, so we talked about those 4 important organic molecules and how to build them. Let's first of all list those 4 important. What are they? Carbs. What else? Lipids. Good. What else? Proteins and what? Nucleic acids. Good. Okay. What are carbs built out of? Yep, sugars. And what are the 2 categories of lipids? Fats and steroids. Now we have a whole, remember that simplified diagram that you all have where I broke it all down? So I would look at that. So what are fats built out of? It's 2 things. Glycerol, good. And what else? Fatty acid, what? Chains. And do you remember those fatty acid chains could be sat or what? Unsat. Good. Steroids are what? 4 carbon rings. Good. What are proteins built out of? Amino acids. And you remember that's a central carbon, right? How many different amino acids do we have?
20.
Why? Different R groups. Good. And then nucleic acids are built out of what? Nucleotides. And nucleotides consist of a base, a sugar, and a what? Phosphate. And we talked about the 5 different bases, the 2 types of sugars, and the 1 phosphate. You have a whole chart with all of that in there, right?
Yeah.
Okay, so if I want to, and we talked, give me the monosaccharides of the sugars.
Glucose, sucrose, and fructose.
Sucrose is not, it's a disaccharide. Sucrose, what is it? Galactose and fructose. And fructose. And if you do 2 glucoses, you will have maltose. Didn't I teach you this?
Yes.
And if you have glucose plus galactose, you'll have lactose. And if you have glucose plus fructose, you'll have sucrose. And those are disaccharides. And these are monosaccharides. Saturated or unsaturated fats, which one do you see with animals?
Saturated.
Yeah, animals, and unsaturated is with plants. Okay. Now, yes.
So, which one's the monomer and which one's the polymer?
These are the monomers. Color. Okay, so these 4 right here are the polymers, and these right here are the monomers. These are the bricks, and these are the walls. Think poly, like mini. Does that make sense? So how do we get from bricks to walls? We do what? Dehydration synthesis or condensation synthesis we build, right? How do we get from walls to bricks? Hydrolysis. So going from here to here, we're doing dehydration synthesis, and going from here to here, we're doing hydrolysis. It's a hideous chart. But are you following it? Okay. Can I turn it? Okay. So here we're seeing building. So we're bringing them together, and you remember we're taking, collectively, we're forming a what? Water molecule to do that. Because we have to create a need. Why should they be together? I have to take something away from each one of them so they'll want to hook up. So then we can join them together. And the opposite of that is hydrolysis. I have to, if I want to break them, I got to bring something in to break them up. So I send a hydrogen here and a oxygen hydrogen here. All of this requires an enzyme to make this work. It's not, I mean, could it happen spontaneously on its own? But yes, but very slowly. So we need an enzyme to help us do that. Okay, so here you can see the substrate here is sucrose. You know what sucrose is made out of. What is it? Glucose plus what? Fructose. Good. And so we're using an enzyme. Look at this. We're bringing water in and breaking them down into their 2 units. Okay, so let's just, hydrocarbons are carbon atoms in covalent bonds with hydrogen. I throw my arms out there, you tell me the answer, okay? Monomers join to form polymers. Good. Hydrolysis is a reaction that breaks down a polymer into monomers and requires the input of water. Good. Dehydration reaction forms a polymer from monomers. So I'm joining. A molecule of water is formed as a result of moving an H and an OH from another monomer. So what kind of bond can form? Covalent bond. These are all covalent bonds that you're forming here. What do you call a molecule that speeds up a chemical reaction? An enzyme. Which polysaccharides are structural? Let's just go through and do all of these. Cellulose is what? Yep, plant walls. And it would be what? Structural, right? Chitin is what? Yeah, fungal walls. And where else? Exoskeletons, I'm hearing that. And it would be what? Structural. Okay? What's glycogen? Yeah, stored energy in what? Animal livers. Good. And lactose is a what? Disaccharide, and it is what? Galactose and what? Glucose. Good. And starch is what? Stored energy in plants. Okay. All right, so we don't need to do that. What type of fat has no double bonds, solid at room temperature, and usually from animals? Saturated. Good. What type of lipid is made of 4 fused carbon rings? Steroids. Good. Okay, and looky here. Here's an amino acid. We already know him. What do you call the specific covalent bond between 2 amino acids? A peptide bond. Good. Don't worry about that. This is a what? I know, a nucleotide. It consists of a base, a what? Sugar, and a phosphate. How many bases are there?
Well, there's 5 to like
5 total. Good, so give them to me. Adenine, which tends to form double bond or 2 hydrogen bonds with who? Thymine. And adenine is 2 rings. It's a purine, and thymine is a 1 ring of pyrimidine. What's another one? Cytosine and guanine, which is also a purine. And then what's another one? Uracil. Okay. And uracil you would find in RNA. The sugar in DNA the sugar is deoxyribose. In RNA the sugar is ribose. All right. Good, good, good. Yes? Oh, you want that back? There you go. Yes.
So it replaces thymine in RNA? Or
Yes. Okay. So wherever you would want to attach a thymine for complementary base pairing, it would be an uracil instead when you're making RNA. Now, RNA is primarily single-stranded, but it's when you're building RNA off of DNA where you would want to, uracil. Okay, did I skip one? I did. Nucleic acids are built out of nucleotides. Amino acids build proteins. 4 rings of carbon build steroids. Sugars like glucose build Carbohydrates, nucleotides build nucleic acids. When you do dehydration synthesis using, we'll just have to say true or false, 2 amino acids, you form a protein. Do we agree with that? Do amino acids build proteins?
Wasn't this on the quiz?
Yeah, and I went over it in the review and then I gave you a quiz over it. Used the very same question.
Yeah.
Yeah. Okay, amino acids, you form a water molecule?
Yes.
Yeah, when you're doing dehydration synthesis, you will always form water because you got to take away so they join. Okay? Fatty acids and glycerol, you form a nucleic acid? No. What would we need to put here? A fat. Good. Okay? When you do dehydration synthesis using polymers, you form monomers? No. This would be, right? Monomer, you form a polymer. Nucleotides to form a nucleic acid? Yes. No. You clearly get a junior. All right. When you do hydrolysis to a fat, when you break a fat down, what are you going to get? You're going to get glycerol. What else? Will you get water?
Yeah.
No, you'll have to use water. Yeah, you use the water. Okay, good. Which of the following is a DNA nucleotide? Now, the way I would differentiate it out, I do the sugar first, right? So what sugar is there in DNA? Okay, so who can I cross out? B, because it has ribose. Okay, who else? C. Who else? D, because it has ribose. Good. So these both have deoxyribose. I'm good with that. Phosphate, I'm good with, right? So then I have to decide, which one do you find in DNA? Yeah, so it's this one, and I'm going to cross this one out. Okay. All right. Cell theory says what? It's the basic unit of life. Okay. Then we went through, and who needs to go? If you need to go, feel free. If you need to go, bye guys. Bye. You guys are staying? Okay, Swedish. Okay. So when we go between prokaryotes and eukaryotes, the big thing you have to remember here is they have no what? No nucleus and no membrane enclosed organelles, right? They don't have any membrane enclosed. Do they have ribosomes? Yes. Do they have a cell membrane? Yes. Do they have DNA? Yes. Where do they keep their DNA? What do you call that region? Not nucleolus, nucleoid. Think oid, schmeary bacteria. Yeah. Okay, nucleoid. Okay. I know, huh? All right. Nucleoid. Okay, which structure is characteristic of prokaryotic cells? Yeah. Nucleoid, right? This is in a eukaryotic cell, and this is in a eukaryotic cell. Do you agree? Okay. So then, when we look at prokaryotic versus eukaryotic cells, you want to know, like, who decides who goes in and out of this cell? The cell membrane does. I agree with you. Who detoxifies?
Smoothie R. Yeah. Wait, no.
Smoothie R does that. Good. Who else? Do we talk about peroxisomes doing that a little bit? Breaking down fatty acids? Okay. Who supports? Cytoskeleton. I don't know where that is on here.
It's bolded.
It's on the
Oh, there we go. Okay, gotcha. Cytoskeleton. Okay, who's the UPS man?
Golgi.
Totally the Golgi. Energy, mitochondria, most definitely. Store, some vacuoles. There's a vacuole. Okay. Protein synthesis, ribosomes, has ribosomes, roughy R, brain, nucleus, digest, wherever a lysosome is. Okay, we good on those? Then we go, okay, what are some things this animal cell has that a plant cell doesn't? Think Disneyland. Yeah, the centrioles, the churros. Okay, so this is in the microtubule organizing center. Animals have it, plant cells don't have it. Yes?
Are they the ones that also help form basal bodies?
Yes. That's what is hypothesized, because the basal bodies have the 9 triplets as well, microtubules. Okay. All right. Plant cells, same idea as we know before, but how are plant cells different? They have a, huge ginormous vacuole and chloroplast and cell wall. So if I ask one of your classes, diagram a simple cell for me, diagram in 5 organelles, and tell me what each do. You could do that, yes?
Yes.
Yes, you could. Okay, yes.
On the chart, it says that green thing up there is the chloroplast.
Oh, this is a chloroplast, and this is a chloroplast, and this is a chloroplast. And there's a chloroplast.
It's called a gran.
What?
A granura? Is that an individual one?
That's in.
No, that's inside.
Okay.
This is the grana. All of these are referred to as the grana, and inside that space is called the stroma. What? The chlorophyll is embedded in those thylakoid membranes that make up the grana. I haven't taught you that yet. Yeah, there's a lot of stuff in there. Yes.
The animal cell doesn't have a vacuole, right? Or it's just.
It does have a vacuole, just not a large central vacuole. Okay, so let's look at a few of these things. Which organelle is the powerhouse of the cell?
Oh, mitochondria.
Total mitochondria. Which organelle determines what goes in and out? Membrane. Which organelle processes, packages, and distributes? Golgi. Which organelle is the stomach of the cell? Lysosome. Supports, protects, and is permeable to gases and water, so they can enter and exit the cell. Cell, where is that? Cell wall. There it is. Okay. Which organelle is a control center? Nucleus. Which organelle compartmentalizes itself? It's called rough if it has ribosome. Where is it? D. Okay, there we go. Which organelle converts light energy into carbohydrates? Hint, photosynthesis. Chloroplast. Which organelle is a multipurpose organelle? Stores, transports, support, turgor pressure on plants. Vacuoles. Which structure is the site of protein synthesis? Ribosomes. Which of the following structures would be found in both plant and animal cells? Would we find centrioles there? No, that would only be in animals. Chloroplast only in
Plants.
cell wall, plants.
Both.
Both. Yay! Permeability. Oh, what are we on now? Chapter 4, Smarty McSmarties. Okay, yes?
How long do you think until we're
We're pretty close.
10 minutes.
7 minutes.
Oh.
Let's see.
Okay.
I know. Now I need to watch my watch, so I do it in 7 minutes. Yes. Okay. So first thing I would go over is you need to know the parts of the cell, and I didn't put a oh, here's one. Okay. The parts of the cell, okay, and what each does. So you have to make sure you know about phospholipids, and phospholipids act as a barrier. Proteins have several functions. Let's list all of the functions. Channels, carriers, cell recognition, receptors, enzymatic. You're so cute. And cell junctions. Do you need me to write that down? You got it? Okay. This is a glycoprotein, and is this one a glycolipid? And that helps with cell recognition. Good. It's part of the extracellular matrix. And then this little guy in there, he is what? Cholesterol. You only find it in animal cells, and it has to do with what? Fluidity. Because if it gets too hot, hello.
Hello.
Just fine. I'm recording this for the last 7 minutes of our review. Okay. Sorry about that extra little bonus text I sent out to the group.
No, that's great. Thank you. I was just going to come and thank you for that.
Yeah. Fluidity. If it gets too hot, it would what? Melt, break apart. Okay, and if it gets too cold, it would tend to freeze. So cholesterol takes care of this. Who helps us with that when we're talking about plants? What helps them?
Oh, the cell wall.
Cell wall. Yay. Okay, did I hit them all? I think I did. So let's talk about the 3 ways we can get through a cell membrane. We can go through the phospholipids, but not if we are large or charged. Okay, So if we're larger charged, one of our options would be using a channel or a carrier. If we're using a channel, we're going with the concentration gradient. We can if we're doing a carrier as well, but usually a carrier you're talking about using some ATP, some energy, and now we're not talking about passive transport. We've now gone into active transport. And then the third way is whole membrane. We can either do what? Endo or exo. Am I doing something funny?
No, that's the one I got wrong.
What?
That's the one I got wrong.
Oh, your flip-flops prevented you? Okay. I hope you're wearing your flip-flops on Monday. That's all I'm saying. And if you're not wearing them, I want you to bring them and set them on the desk so we can look at them. Okay, I think that's easy done. All right. Sorry, we're tired. You know, just bear with us. Okay, so we did that. Okay, now let's talk about here. If we're using water, if water is going through that phospholipid, it could go through, but it is polar, so what facilitates the process of water going through a cell membrane? It is called osmosis, but what facilitates it is aquaporins. Yeah. And passive, no energy. Active, it does require energy. So let's focus on the diffusion of water across a semi-permeable membrane, and that is called what? Osmosis. So when we talk about osmosis, if we are looking, let me see, I think I have it. I do. Okay. When we talk about water being more concentrated, and this is side A and this is side B, where is water more concentrated? Water is more concentrated on side A because it's water and some stuff, but on side B there's water and a lot of stuff. The stuff we referred to, we looked solvent and solute. The stuff would be the solute. Water would be the solvent. So where do we have more stuff, side A or side B?
B.
Side B, so we're going to call that the hypertonic side. But if you call this side the hypertonic, then by default, the other side has to be a hypotonic. Okay, you can't have a beaker, okay, with stuff in it, and you just say, oh, that's hypertonic, okay? Because it's in isolation. It has to be in relationship to something else. So there has to be some sort of barrier, and you say, this is the hypertonic side, and this side over here is hypo. If it's same on either side, we say it's isotonic. Yes?
So on the quiz, I got one wrong. It was like talking about osmosis and how it's the diffusion of
Was
water
it the pickles one?
in a semi-permeable membrane. What's the difference between diffusion and osmosis?
Okay. Diffusion could be if I put perfume on my arm right now and it started going that direction towards you, it's diffusing, going from high to low.
Okay.
Sodium on one side of a membrane going to the other side, high to low, that's diffusion. When you talk specifically about the diffusion of water across a membrane, you're discussing osmosis.
Okay.
And when you're discussing osmosis, that's when you talk about hyper, hypo, and isotonic, because that has a membrane, right? That's why those words come into play. All right, so now let's do some questions here. This one, carrier proteins are what in their actions? Specific. Cholesterol is found in the plasma membrane of animal cells. Muy bueno. Oh, let's look at these pictures. Isotonic means equal, so the same amount going in is going out. Same thing here too, yes? Okay, in this situation it's hypotonic. The solution is hypo, so inside it has to be what? Hyper. Water must flow from the hypo and it causes it to swell. The animal cell won't burst, the plant cell won't burst because it has a cell wall and the turgor pressure, the wall will push back. But the animal cell, can it burst? Yes. All right, hypertonic. In that situation, if it's outside's hyper, then the inside must be what? Hypo. Water must flow. It flows out and it causes them to shrivel up. Good? Okay. Let's look at this. When mixed together, water is the solvent. If we know that, then who can we cross out? C and A, right? Because we're saying, all right, water is a solvent. Do this. Obviously, you can't write on my test, and I know that's annoying, but you could do something on a backup sheet so you don't get confused. Like think, okay, those and those are out of the question. Now I'm just looking at these two. That'll just help you so you don't get confused. Water is a solvent, and dye must be the solute. Because solvent, that's dumb because you don't have that, right? That's the one I wanted. The hypotonic solution contains the most solutes. False. We would say the hypertonic. The hypertonic contains the most. Okay? So when a cell is placed, and in this, anything I ask you here, I would make sure, I get lots of visitors, I would make sure that I drew it out on my scratch paper. Okay? So I'd go, okay, the solution is here, it's hyper, and you write things like this is hyper, and then this is hypo. What's going to happen? I would envision in my head, water's going to come out of it, okay? So then I start looking at it. The solute exits the cell? No, because the solute would want to enter it. So I don't even care what else comes next, because that's just wrong. Water exits the cell towards an area of higher solute concentration? Yes. Okay, because all this stuff out here is solute. That's what's making it hyper. Okay, water enters the cell towards an area of no solute and no. Okay, so strategize by making drawings. Okay, then the last thing was the endo and the exocytosis. And endocytosis, we had 3 types. What are the 3 types? Pino, phago, and what? Receptor mediated. Perfect. Pino is cell drinking, I can't write. Phago is cell eating, and then this is where it binds on, right? And it's a type of pinocytosis, but just a little more specific and a little more efficient. Exo is anytime you go out, right? So here was that receptor-mediated endocytosis, and here is a question. This is what? Exocytosis, because it's what? Going out. Exiting. Which of these methods are endocytosis? Is A? B? C? Swedish. Pumps used in active transport are made of proteins, because you would know, right? Pumps are proteins. It's an active transporting protein. And pickles appear wrinkled and shriveled because they have been placed in a hypertonic. Water must flow from them, and they get smaller. Yay! Okay, don't stay up too late. Make good choices or have some toast. I always say have toast.